Good morning, everyone. Thank you for the organizer to give me this opportunity to share our work using LabKey Biologics to map our cell line development workflows. Thanks here. To start my talk with a brief introduction of the cell and developability sciences group and our cell line selection process. Next, I will describe how we use LabKey Biologics to map the COD process, which include how we register the molecules, how we track the lineage, as well as some other, how we associate the assay data with the sample, as well as organize the samples and assays with experiment. I ended my talk with talking about some challenges we're facing, including how to create the unique IDs, how to use it in our daily workflow, as well as some automation and project-centric challenges. So first, I'll give you a brief introduction on our group, which is Cell and Developability Sciences. As you can see in these slides, our group sits at the beginning of the development of large molecule development. So once the target is identified at Janssen, we screen from hundreds to thousands molecules. That will go through a series of steps, include hit-to-lead generation, lead optimization, that narrow down the candidate molecule to 1 to 4 molecules. And it's our job to take those molecules and transfect them into cell line and generate a manufacturing cell line. We also have analytical support group, like doing a series of biophysical as well as mass spectrometry analytical assays on those cell lines as well as the product generated from the cell line to ensure the cell line has the best quality. Over the years, we have been seeing we're shifting for the large molecule development. We're shifting from pure antibody platform to our platform program. To give you some examples, some multi-specific become very popular. They are not just tracking one target, but also recruiting T cells at the same time. We also see a lot of vaccine type of work. Those type of programs present a new challenge, not only for the cell line development process, but also for the analytical challenges, different assays, different properties we have to develop. It's not surprising we're constantly in the battle of taking more molecules, which means not only pure number of molecules going through generate the cell line, but also different mechanism of actions. We can test more different mechanism, so we can bring, hopefully, but at the same time, we also want to reduce time to bring those products to the patient earlier. But how do we do that when we have those requests? So the first, this is our typical cell line development process, which we doing a transfection and go through 2 stages of a cloning process, parental cloning and subcloning. And as a group of 8 or 10 clones were selected from this and move into amber bioreactors and generate a large amount of material so we can do analytical analysis on those samples. That will guide us how to select the manufacturing cell lines. Over the years, I think in the last 2 years, we've been developing a new process trying to shorten this timeline, particularly totally remove one stage of cloning, but a subcloning process. But at the same time, we're not we still ensure the clonality of the clone, monoclonality of the cell line with selection. But by doing this, we're eliminating our timeline by 1.5 months. Those technology also have some other advantages, like it's a site-directed integration. We know where the plasmid will integrate into the genome, and so which makes the titer more predictable. That also allow us to screen fewer clones, shorten the timeline. On the other hand, we also generate this particular early look material from transfection pool, so we can start analytical method development sooner. So the analytical method analysis won't be the bottleneck of this whole process. So here I showed one solution in cell line development to shorten the timeline, which is over the years we have been reduced the timeline from 7 months to 5 months, but we still have another hurdle to overcome, which is the information management. That was estimated like every scientist will spend 2 to 4 hours per week to gather information, to find the information they need, because the information all spread along or sitting by email. And so how do we hurdle that? That's because those time, I wouldn't say wasted, but spent on this kind of analysis adds up. When you consider we have 30 scientists in the group, and that's over 6,000 FT hours per year. So the information, where are the information sitting now? We have an electronic laboratory notebook, but in those notebooks, we generally save the data in different formats, as you can imagine. Some assay data or sample submission in Excel sheet, and some of the results are summarized in the PowerPoint slides, and some protocol or other documents in the Word and PDF. They all scatter around. They're not lost anywhere, but to putting them together, generate a meaningful report does take time, especially when you go through several rounds of screening, and when you find your final clones, you want to find where the parents are, what is the information associated with those parents. That becomes challenging, especially when you have multiple scientists working on the same project. So that's where the LabKey come in. We use the specific product we use is LabKey Biologics. If you not have seen this product yet, this is the landing page of the Biologics. Has 6 different functionalities: registry, assay, sample, media, workflow, as well as experiments. And today the medium and workflow I won't be talking about because we are not spending enough time to developing the functionality in those areas yet. So, but I will touch briefly on each of those 4 one by one. The first one is registry. First, we need to know what the molecules coming into our shop. So the first thing we want to know, we have if you remember the previous slides, we screen from thousands of molecules, those through heat to lead or co-optimization and narrow down to 1 to 4. But we don't want to. We have a system to store those information, but we don't want to just dump all the information into LabKey. We want to select the ones we need that come to our shop. That's why we developed a Python script with the help of Susan in LabKey. And basically we select, give a unique identifier, select the molecule, and the script will automatically extract the data, export the data, basically the sequence, the protein and DNA sequence, and as well as some metadata associated with them, and import automatically into our LabKey Biologics system. By doing that, we have a list of molecules that have associated DNA numbers, some other with correct components, like, for example, those are antibodies. We have light chain and heavy chain, and as well as same time when it's starting, LabKey automatically calculate the mass and some biophysical properties of these molecules, which is make it easier to use. So then I would like to dive a little bit detail into our cell line development process, because that will help us to understand why we need to track the lineage and why it's become challenging to associate all the data comes with this analysis. So first we got a plasmid from our upstream folks, and we transfect into the cell, but the cell pool, transfecting pool, would split into multiple 96 deep well. And based on the titer and other information, we narrow it down to 24 deep well and eventually to shake flag. That's the parental cloning stage. And from that, individual clones were going through another round of selection, which is another 96 deep well, 24 deep well shake flask, then bioreactor. And 8 to 12 of them will go into bioreactor, generate larger quantity of material for the bioanalytical analysis, which will guide us to select the best cell line. And those are the data, just to name a few, like associated with different steps. For example, we have some white cell data associated, which means the how many live cells in the culture, and the titer data, how much the material was expressed in the cell. And as you can see, when you have multiple plates, multiple samples, multi-project, it's become challenging to organize all this data. And here's LabKey, how LabKey can do this. In this example, we have one cell line selection project. We did 2 transpections and pulled them together, and that will split into 208A, which is 4 different plates, 496 deep well plates. And as you can see here, not every sample in the well goes to the next step, but some does go down and to the subcloning stage, which will do another splitting into 96 deep well, those the subclone, and eventually go to a late stage. This is how we will be able to track the whole process in the LabKey system. The next functionality I want to describe is the assay. So we were able to create an assay format for a series analysis. For example, the octet titer, protein A titer, as well as some biophysical experiment like CIEF, SEC, and also some peptide mapping like mass spec type of data. Here I show you one example of the assay we imported. It's called the reduced mass analysis. This analysis, if you're not familiar with, it tracks the glycosylation distribution of a particular antibody product. The antibody are generally modified, by different glycosylation. But tracking this glycosylation profile is an important step in our cell scene selection process. By putting this together, which I can show you in this slide, so we're not only just looking one molecule across different clones, we're seeing different glycosylation profile, we're also able to compare 2 different molecules which target the same target. So that become reasonable or doable, which after we have LabKey system. It used to have, you need to just to spend a lot of time gathering the information and put them together, which LabKey help us to make it easier. So now we have a lot of samples, and we have assays associated with samples. But if we're particularly interesting at today's stage, especially the cell line selection, when we have 8 to 12 clones, we need to group those clones together and look for only the assays associated with those clones. That's where the experiment functionality come in. So here I show the example. I have 2 different set of samples. One set was sent to biophysical assays, doing SEC or IEF type of work. The other set was sent to us to do mass spectrometry type of analysis. By associating them into one experiment, we were able to bring all the data together on this page. So you can see we have different assays, reduced SDS, non-reduced SDS. We don't have mass spec, but But you get the idea, like basically that group everything together, filter down to the assay for the experiment very interesting. That's how we gather the experiment together. So that's the stage we're currently in, and the last part of my talk, I want to talk about a few challenges we're facing while implementing this system into our daily operation. The first question is, it's the sample ID and the lineage. How do we track this lineage? Basically, the most general cases we're getting information is from the assay results. But how do we incorporate the parent information into the assay data file? Can we do that? If we can't do that from the input in the instrument or assay instrument, can we do that some other way? That's the if. And when we import those data, are those parent information already exist in the database? If not, can we create those? Those are the questions we're constantly asking. Another question is, so how we make this ID easy to use? Because LabKey can easily generate 100,000 IDs, but it's just a number. And maybe you can design some tags into it, but how do we make it easier for the scientist to use, putting it on their like bioreactor, on their tubes plate? That's become the challenge for us. So we're not trying to, like, we generated 96 well plate, it's the number 1, and that number 11 goes to the next stage, which is the number 21, or number 1023. That's not practical and is error-prone for the scientists to use in their daily operation. And that does require the scientists to change their habit a little bit. And remember, we're not starting everything from scratch. That's what we decided. This is what we're going to do. People already have their way of doing it, no matter how inefficient you might think, but that's what their way, that's a comfort zone they're used to work with. Making this change is always challenging. Another challenge we have is the automation. So this is one example, and one of the peptide mapping assay, just one type of assay we're having is to look at the post-translational modification percentage, percentage across different clones. But in this case, this software export first, although it's a CSV file, but it's not a regular. But you can see this one, the header is not on the first line. So there's some format we need to change. At the same time, this looks 8 to 12 different clones. This column sometimes has 8 columns, sometimes has 12 columns, but it's referred to different clones. To be able to transform this kind of result to a different format, like basically compile this 8 columns into 8 different rows for each peptide. With the help of HANA, we were able to do that. So using the tools LabKey transformations and script, we're able to transform the data from one format to the other to allow better organization in the database. Another one is always our end goal, is trying to encrypt all the different instruments in the lab, trying to get the data directly, but also link to how do we let the system know where the sample come from, where the lineage come from, or what is the information we're extracting from this. It's easy to see. We say we have we can export and import all the information, but that probably also creates some chaos in the system, and might not be easy to parse at the end. Another question is how should we do that? Every instrument has different format, and it requires individual attention to export or maybe link to the system to LabKey. But this is our end goal that we're trying to work on. The last maybe a suggestion or our proposal would be as we work from project to project, which means one molecule, like one target to another. We also have our COD campaign. So that campaign is basically defined. We go through multiple steps, either, in this case, I show as parental and subcloning stage. But either one, even we just remove the subcloning, we still have defined steps. And all those samples associated with those steps as well as assay. Is that possible to create a separate project, and we can, when we have the assay data, we can directly upload to that specific step. So that may be able to eliminate the requirement to name some of the parental or ancestor information. With that, I think it's our end goal or ultimate goal to build the business intelligence in our COD process. Basically, with more efficient, first, it's more efficient clone selection process because we can track the cell line development, we can integrate all the data together, as well as generate the report automatically. So that will help us to make a more informed decision because the data integrity, and we'll be able to use with these tools, not with the electronic laboratory notebook, we will be able to compile all the data together, doing some artificial intelligence or machine learning, and to be able to help us to drive our decision, make smarter decision. At the end, I would like to thank my supervisor, Hershnander, and my colleague at Cell Line Development Group, AppG, as well as get constant support from LabKey folks, Hannah, Kevin, Susan, and Ryan. With that, thank you for your attention, and happy to answer any questions.